All right, I'm going to be talking about my work at NC State, which was on potatoes. Might have been confused by the flyer, seeing a Cotton Lab person talk about potatoes. I do have a legal background in it. So, anyway, our project goals for my project at NC State was to utilize modern genetic and genomic tools available to potato breeders to address agronomic traits, internal heat necrosis, by creating linkage map and performing a QTL analysis. And well, this had been done before. There's been a lot of QTL analysis out there. In fact, my population that I analyzed had actually been already analyzed previously by Per McCorn, using predominantly AFLP markers with a few SSR markers to anchor those linkage groups to the published chromosomes at the time. And in his map, he had 274 markers for Atlantic, and the 1829-5 had 252 markers. Those were the parents for the diagonal cross. So why do it again? And to address that, we need to look a little bit into autotetraploids, for those of you who have never worked with it. And diploids, it's a little bit more simple. You have two possible chromosomes that you inherit from each parent. 1 and 2 from parent 1, 3 and 4 from parent 2, and you have 4 possible outcomes. An autotetraploid is a bit more complicated. For the same, for one locus, you have 36 different possible genotypes that you can get. And then you add recombination to that, things get messy. So one of the problems we have with the old marker system, which was AFLPs, its presence absence, is you look for a band. So say you're tracking the B allele here, you're running the gel, you're looking at it, oh, you got a band for both parents and then for all the progeny. Well, what does that tell us? It doesn't really tell us too much. It's not polymorphic, we can't really extract too much information from it. In fact, you miss a lot of cross types as a result of using the method that had been used previously. And you really don't get any segregation amongst the dosage of alleles, you just get one band. And you can look at it and you can say, okay, well, I have a 5-to-1 segregation in the progeny, and I had one parent had a band, that band must have 2, but as you can see, you still miss out on a lot of information. So that brings me back to why we wanted to do it again. Since the publication of that last paper, a lot of stuff has been done. We published the sequence of the potato genome, and we had the development of the 8303 SNP potato array. This is actually a picture of it right here. Each one of those little squares has 10,000 markers on it, and this little chip runs 24 different samples. And since we are able to use SNPs, codominant markers, new analytical techniques were also developed to utilize that information. So essentially how it works is you got this chip right here, and you run your samples, it goes in here and laser excited for each SNP, and it produces 2 different colored fluorescents. And then that machine can also read the ratio of those 2 different colored fluorescents. So if you're looking right here, this is an example of one locus. In both parents, you have this cross. You should have, 50% of the illumination being one color as well as 50% being the other color. And it should segregate in this way. For this type of cross, you would have a 1:8 to 18 to 8:1 ratio. And the ratio of those illuminations is called a theta value. So then you can plot those out, and this is an actual marker from my study for one of these. You see the 2 yellow dots are the parents, which would represent an AABB by AABB cross. And there you have each cluster for each ratio there. And if you do the math on that, it's really quite close to a 1 to 8 to 18 to 8 to 1 ratio. So we're able to get a lot more information from each of our markers this way. And as you can see, it really opens up this table. And not only do you just have a band reading, you can see the dosage of each allele. And that really helps you in combination functions, as well as performing phasing. You can get a lot more precision and accuracy. So what does this mean? Well, we have the better SNPs. First of all, it's a codominant marker instead of a dominant marker. good. SNPs are genome-wide. We can analyze both parents simultaneously, whereas before you had to create the Atlantic map and then create the B1829-5 map separately. We could put them up line by line and go at it. Sorry. So we can begin to use this information to put together a better picture of what is going on with these traits. So for materials and methods, like I said before, we used a cross that had been used in the previous study. And the reason we did is because Atlantic is still a very, very strong variety that's grown throughout the United States for chipping. North Carolina is a major chipping state. That's mainly all we produce our potatoes for, is the processing industry. And B1829-5 was a nice clone that was developed out of Beltsville, Maryland. This is IHN, internal heat necrosis susceptible, which is one of the components we wanted to look at. And then this is pretty resistant to IHN. So in that cross, we had 160 progeny. It was evaluated over 4 seasons from 2006 to 2008, and I gave it another go in 2014. In the first year, it was an unreplicated design with 6 plants per plot, but then we got enough materials generated, we were able to do our CBDs in 2007, 8, and 14. So I'm going to separate here and look at the agronomic traits first, and then we'll come back to the internal heat necrosis.
Which generation is the progeny?
F1.
F1, okay.
There's no inbreds in potato, it's now crossing crop.
Yeah, it should be like F2.
Well, there's no inbreeding in potato.
Yeah.
So we did the F1, and everything was heterozygous in both parents. So that's why we genotyped both parents also, so we can get that information for creation of maps. But anyway, I'm going to separate and to go to agronomic traits, and then we're going to go into internal heat necrosis. And the reason I did that is because there was a lot more work done in the agronomic traits. So I wanted to analyze those first to kind of get a good idea of how good our map was, because there had been QTL identified in the past. So for phenotyping, 2006, 2008, obviously it was done by my predecessor, but essentially all tubers were washed and culled for excessive rot, and we looked at dry matters, specific gravity, yield, skin texture, and foliage maturity. So for dry matter, we just took 4 tubers from each plot, we cut them in quarters, we bagged them and weighed them, and then we freeze-dried them and weighed them, and it was just this formula, dry versus fresh weight. And for specific gravity, it was the same thing. These are essentially believed to be the exact same thing, just different methods of taking it. The reason these traits are so important is for processing quality. It gives you a nice crispy potato chip. And then we looked at yield, which is we just calculated as the total weight minus the tubers culled. In 2006 through 8, it was done by per plant place. It was done over plots and then divided by the stand count, so you can get a yield per plant kind of rating. I didn't do that. I just took the overall weight of the plots. Skin texture, we scaled on a 0 to 3 in half-point increments. And then foliage maturity, I took ratings at multiple times, and then I graded it on a scale of 0 to 100. Percent senesced, which is a 5, 0 for 0. And I took the ratings 3 different times, and then I plotted those points out, and I took the area under the curve, and that area under the curve was the rating I used in my analysis. So for genotyping, we just collected tissue samples, DNA extracted using CTAB, and I was very fortunate to be able to send it off to MSU for their pipeline to have it genotyped. So for creation of the map, I took all 8,303 markers, and I took those theta values and I used a multiple mixture model to get them in each of their clusters, and that's how I was able to determine the allelic dosage. From there, I removed all markers that weren't polymorphic. I genotyped both parents twice because the parental information is so important in creation of the map. And then those that the parental information didn't match up in both parents or in both replications, I went ahead and tossed out that marker. And then I removed markers with 10% no-call progeny. So if during those multiple mixture models, one of those progeny didn't get a theta value that clustered very well. We had high confidence with, in 16 or more of my progeny, then I went ahead and just threw that marker out. And then I inputted the markers into tetraploid map. When I say tetraploid map really doesn't exist for this type of analysis. We were in the process of creating it. I was working with the bioinformatics lab in Scotland, and we were kind of going back and forth and using combinations of R and standalone ISML libraries. And it was very tedious. And then I double checked my map once I was finished by running the QTL analysis on those theta values that I originally developed, and that helped me establish if my phasing was right, if my positioning was correct. And then if it wasn't, I threw those markers out and I went back and I did it again. So I did this a few times. It was a tedious process, but in the end we actually got a map with a pretty decent amount of markers. We had 3,427 SNPs at the end of the process. And we created a pretty dense map. As you can see, we had a lot of markers on each chromosome. Length was from around 98 to 139 there. Overall map length, 1,397 centimorgans. These max distances, these are standalone on each chromosome. The average distances, as you can see, was actually about a half a centimorgan. And then the max distance was kind of an isolated event, and we assume that those are the centromere locations where those max distances are. So anyway, we performed the QTL analysis using these components of tetraploid map. It uses an interval mapping method using an additive model, and then at that location, it will spit out the output of the hidden Markov at that, which gives us the 36 possible genotype means at those log scores at that locus. So then I took those 36 genotype means out and I looked for a simpler model if something seemed out of order. And essentially what I did is say I said, okay, what if the QTL at this locus is only on chromosome 1, homologous chromosome 1? So I took all the genotypes out of those 36 genotype means that had contained homologous chromosome 1. And I did a simple t-test versus those who didn't. And I did this for simplex by nullplex, simplex by duplex, and simplex by simplex marker crosses. You can't really do much more than that because you're only working with 36 values. Now these 36 values, each one of them was created from the original 160 progeny, but still it gets a little reaching once you go beyond that. But it still gives you a good idea of what to look at and if anything kind of suspicious is going on. So y'all have seen distributions of traits before. As you can see, pretty much everything was normally distributed. I had to do yield over two different distributions per plant. I did overall plot. And the same thing for maturity, frequency distribution for maturity. The reason I did second distribution here is because I did it at different dates, so the number scale was shifted as a result of my area under the curve measurement.
Mitchell,
Yes.
I'm a little confused. If you go back to your last one, so I mean, if you're saying that's per plant, you know how many plants, so why couldn't you make them the same?
Because we didn't do the stand count this year.
Yeah, but couldn't you take the grams per plant and multiply the number of plants? Because they knew the number of plants, right?
Yeah, I guess I could have. I didn't do that. But, oh, I couldn't find the stand count data. That's why. That's right. So anyway, texture was kind of all over the place here, but it was really hard to analyze, and there's a lot of different things that we think are affecting that.
What is your X values on your X-axis?
For what?
For any of these, go 0 to 150 for the upper right.
Oh, for the maturity, that is
days?
That is the value we calculated as the area under the curve. So we took the maturity ratings on a 0 to 100% senescence, and we plotted those, and we just took the area under that line that was developed.
So the higher the number, the later maturing.
Yes. So, for the QTL analysis, what you're looking at here is a whole genome log profile. Here's your log scores on the y-axis. On the x-axis is my map. And each of these values is the log score from the association of each interval. So the black line here is the 95% confidence interval. The gray line is the 90% confidence interval. As you can see, these lines were developed by a permutation test. So we did 500 permutations, and we kept running the analysis each time we permutated the data. We did that 500 times to establish these values. As you can see here, we got two different yields that cross those lines on chromosome 5, and then we had a little one here in chromosome 6, and another one in chromosome 12 for yield. But what we really were interested was we had two different years in the same location that were significant. Again, for skin texture, we just had this little peak. We had multiple years peaking here on chromosome 4, also a little bit in 5, and then kind of a jumbled mess here on 9, but it surpassed the threshold as determined by permutation score. This was our largest QTL, as you can see by the log scores here, and that was on chromosome 5, and each year that we analyzed it, right there, had a very significant peak. And then we had chromosome 7 also. We had 7 and 14 had significant log scores. For dry matter, it was dry matter and specific gravity. I'll get into that in a second. But as you can see, we had two peaks here, but they weren't really close together. And then we had a peak here on chromosome 11. And then specific gravity, we had multiple peaks here on chromosome 5, and then a little bit going on here on 8 and on 10. We were kind of interested to see that none of the peaks for specific gravity and dry matter co-located. And we expected to see this co-location because a lot of times, because dry matter is more complicated to take, we just take specific gravity as a rating for the dry matter qualities, for processing qualities. And as you can see, we did have correlation between the traits. The published literature has a lot higher correlation. There's a lot of things that affect it. Environment might be one. We have a lot of rain late in the season where we do this. But we still did have a lot of correlation. So what I did is I went ahead and lined up the log profiles. And you can see here that the lines really do kind of follow each other, and the correlation is very comparable. So we believe there's still, even though they didn't say, like right here, this peak for specific gravity was significant, but there was a little peak here, but it didn't quite get to a significant level as determined by permutation there on chromosome 8 for dry matter. However, the lines follow each other quite well. So we believe there's a similar genetic background to this trait, even though it didn't get to those established marks by the permutation test. And so, moving on. One of the good things about this is we were able to confirm a lot of these previous QTLs from other studies, which helped us see that our map was a pretty decent map and was lining up with other things in the literature. Another thing we found was chromosome 5 was a huge hotspot for us. If you go back and we go through all those log charts again, I'm just kind of breezing through them. But we had a lot of stuff that hit chromosome 5, and a lot of stuff had its most significant log scores on chromosome 5. And it was the only location where yield at the same relatively close positioning had significant log scores that overlapped in significant ranges. And if you look at right here, this big blue line was the largest yield of QTL, and then I overlapped all the other traits here that had significant lab scores on chromosome 5, and it all happened within that range of yield, which kind of begins to make sense, especially when you go into the literature and you read that late blight resistance, potato virus attacks, tuberization, and as you'll see in a second, internal heat necrosis, have a lot of significant QTLs on chromosome 5. So plus all these agronomic traits that we found, dry matter, maturity, yield, 2 yield hits, there's definitely a hot spot going on here on chromosome 5 that would be of interest in the future. So there's still a lot of work to be done with these agronomic traits. This is still a relatively new tool for our community. The SNP chip just came out when I got there. It was just developed. We're moving in the right direction. There's only been one other linkage map in QTL analysis published to date using this technology. And as we're developing this, we're expanding our database information. We have a database called SpudDB. We're updating that information. So we had overlapping traits with this study, which was maturity. And one of the interesting things is I looked up their marker that associated, that they proposed as a candidate SNP in their paper. And it was really, their marker did not segregate in my progeny for one reason or another. But a marker that was closest to my largest significant point was very close to theirs and very close to a reported transcription factor that does affect maturity as well as tuberization. So that also helped us validate our maps, but also we can begin to compare information between these studies by using these SNP markers, whereas we couldn't really do that before. We can really start to build up our database. So now we're going to move on to internal heat necrosis. Internal heat necrosis is a non-pathogenic necrosis found in potatoes. North Carolina, it's responsible every year for 3 to 11% of crops in the field that just don't even get harvested. If you have 6% in your load that has IHN incidence, then you throw out the entire load. They won't accept it at the processing facilities. And they say that there are some acrylamide issues with it, but mainly it's just an ugly potato chip, and people don't like ugly potato chips. It's all skipping. So what affects IHN that we do know is high day and high night temperatures. That's why we call it heat necrosis. But also there is correlations reported with calcium levels in tubers for internal brown spot, which is something they see in different areas. We believe these things to be the exact same thing, internal brown spot, internal heat necrosis. So we use the same population again. I won't go into that again. The way we graded this is we actually gave it 2 different ratings. We gave it a severity rating. So what we did is we took 20 tubers from each plot, we quartered them, and then I rated them on a scale of 1 to 9 for severity, all 20 tubers, 9 being no necrosis, 1 being completely necrotic. And I took the average of those severity ratings, and that was my severity rating. And also for incidence, the percentage of those 20 tubers that had any sign of IHN. That was my second rating for IHN. So if you look at it, it's highly skewed in all years, significant year effects on population mean. And the same thing for the other one, it's just worse because the scale is different. But it's still very highly skewed. In fact, these traits are very, very strongly negative correlated, which we expected. We're using 2 different terms to describe the exact same thing. Another thing is when you plot the residuals, it's very, very homoscedastic, or not very homoscedastic, sorry. If you look around here at the 9, resistant is pretty fairly consistently resistant, and then it's all over the place for the other ratings. And that makes sense a little bit because there's so much environmental influence on this trait. It depends on temperature, it depends on rainfall, and it needs a trigger. So when we did the log profile here, same thing we were looking at earlier, we really only had 2 locations. This got above the 90 percentile, this got above the 95 as according to the permutation test, and chromosome 5 and 1. So we really didn't get a lot of information here, and we're hoping for more to look at it. So I started to think, well, what is the purpose of the permutation test? and it's to help eliminate type 1 errors. And so what are some of the characteristics of a type 1 error? Is they're random and independent. So we started talking and we're like, well, we have multiple years of data that we can look at. And if we look at these points and they have peaks at the exact same location that are clearly defined throughout the noise, then that kind of violates the random assumption of the type 1 error. So we started looking at those peaks on here. We found in chromosome 9 and 12, although it didn't get past the permutation threshold line. Another thing about these permutation threshold lines is they're developed, how high they are is based on a genome size also, and our map was so much larger than other maps. A lot of people in the previous published studies used a log significant threshold of around 3, but ours was so much higher. So we looked at these points on 9 and 12 where we had multiple years forming defined peaks at the exact same locus. You can see maybe chromosome 3 here, but these are the exact same years that form these peaks. So because the traits are so highly correlated, we would, and they're described in the same year, we didn't take those points. But we did look further into 9 and 12. So then again, we looked at the 36 genotypic means, and we fit simpler models, and I did this for all possible arrangements, and then I used a Schwartz information criterion to look at each model to see which one is the most parsimonious or the best fit model. This slide is to kind of show you what I did next. I'm just going to use this slide as an explanation of what's going on in the next slide. So this is the genotypic effects of those 36. So here's the 36 genotypes, possible genotypes. And I took all the genotypes that didn't have homologous chromosome 1 or 5. Remember, you get 1 through 4 on parent 1, 5 through 8 on parent 2. And I went ahead and see which genotypes didn't have a 1 or a 5, which are these genotypes. And I plotted those out for their incidence scores. And I did that for 1 and 5 because the model that had the best fit that I looked at earlier using 36 genotype means showed me that a model with chromosomes 1 and 5 explained the most amount of variation. And what I did here is just kind of to better visualize it, because you see there's a bunch of gaps here and jumping around, which is consistent with our data, which was jumping around once you get into resistant lines. You see there's a lot less variation here amongst resistant genotypes between the susceptible genotypes. So what I did is to kind of even that out across, and so I can compare the multiple years that had those peaks, is I added them all together and I just simply took these values divided by that total, and so I got so it gave me essentially an average effect of these genotypes, or the average amount of incidence that contain these genotypes out of the total incidence, which was 10% here, and then 21% there, and then 27% there, and 42% there. And mainly I did that so I could look at all these at once. And if you look at the incidence and the severity on the right, you look at these pie charts, they're very identical. So it makes me think, and it's through multiple years, so it makes me think that maybe this is the best model in our population for these genetic effects, is we're getting a QTL that's coming from homologous chromosome 1 and 5 that's causing this susceptibility. So what I did is I went back into my map and I looked for a marker within that significant range that had polymorphism on homologous chromosome 1 and 5, and I just tested that, because in the QTL analysis we tested it for 1 year. I tested it across all 4 years and considered year effects, and it was very significant, and you can see that it seemed to have an additive effect on it. This is 0 dose. This didn't have the 1 or 5. This had either 1 or 5, and this had both 1 and 5. So then I put it all together. I did this for each position where we found potential QTL, and I mapped it all out. Or I did it, and I did that. I found a marker for each one, and I put it together in an effects model to determine for overall years, so for all 4 years that we took the data. And I found that these 4 markers explain 28% of variation, which isn't bad for something that's so variable based on environment. One of the things you might notice is these 2 markers, which is on chromosome 9 and chromosome 12, they modeled best as a dominant marker. And if you go to remember the QTL analysis was performed using an additive model. And so that may be why these weren't significant, even though in this model, coding them as dominant, they have the most significant, or the highest F values. And we think the reason is because obviously the degrees of freedoms are different. So maybe in the future, an analysis that includes both additive as well as dominant effects may be best. So going back to what we know in the literature, that there's a role of calcium, that tubers that have a higher amount of calcium correlate with IHN or IBS resistance. So I found a PhD dissertation out of Wisconsin that just came out, and I believe they're working on the publications for this now, where he found SSRs from a CAX3-like genes in the potato genome, and they were shown to, he developed these SSRs out of that gene and ran them in the population and found that they associated with calcium content in the tubers. And so what CAX3 does in Arabidopsis is it's known for calcium transport, and it's highly expressed in the roots and flowers. So I went through and I blasted the potato genome for these CAX3, for the CAX3 sequence, and I found it was very similar to a vacuolar cation protein exchanger 1A, very similar. It had a nice low E value, and interesting enough, these were very close to the markers that I pulled, but within still the significant range of the QTL that we identified. And one of the good things is this, I didn't say this earlier, but the SNPs that were used in the SNP chip were developed from 69,000 high-quality SNPs in another study, all found in the EST database. So we actually have information on a lot of these SNPs. And we found this SNP, one of the SNPs that we used in the analysis was found inside the vacuolar proton cation exchanger 1A. So, and we also found that this marker was in phase with our best fit model, except for the addition of a homologous chromosome 1, but remember we didn't test that, we only test simplex and duplex. So another thing we found is when we analyzed that marker with all 4 years of data, it explained 10.2% of variation. And so essentially it was just came down to 3 copies of one version of the SNP. The mean of those genotypes was 5.5%, and all cytidine, because the way it's segregated, was 20.7%. So we found that it did have an effect on meat. It was very significant and explained 10.2% of the variation. So maybe it's a possible candidate gene. Obviously, a lot has to be done, but we're starting to build a case for it. We found that it supports the literature, the research we've done in the literature, that calcium correlates with IHN resistance, and these CAX3s associate with calcium levels in the tubers. We found that the CAX3 homolog co-locates with our identified QTL in the study, and so that is in phase with our best fit model that we found in the study for that particular QTL. And that marker is highly associated in this study with IHN susceptibility. So more work would need to be done, but it's definitely a direction to move forward. So in summary, through this analysis, we found 20 putative QTL for agronomic traits. We found 4 for QTL for internal necrosis. We started working on modeling genetic effects of each QTL, and we identified markers that we can actually use in that genome database for future studies and references that were co-locations and also strongly associated with those QTLs. So anybody who does a study later can go back and look at that. So and we identified the potential candidate gene for future research. So where we are, as I said earlier, we got these new resources, we're working with them, we're learning how to use them, but we're heading in the right directions. We're building a solid molecular genetics and bioinformatics infrastructure. There's still a lot of progress to be made because this is new to our community, but I think we're definitely moving in the right direction, and we're able to utilize these resources more efficiently. So anyway, here's thanks to everybody, my committee members, my advisor, the research tech, as well as all the financial sponsors for the research.
Mitch, does internal heat necrosis affect the nutritional quality of the potato chip?
Yes, we do see higher acrylamide levels, which is now a hot topic issue in potato. It's a carcinogen. However, there's more acrylamides in coffee, but it's definitely affecting the community, and they're putting a lot of resources into removing the acrylamides from potato chips.
You had 160 progenies. What was the range of maturity among them? Earliest and latest in number of days or whatever.
It wasn't too much variation there because these were more advanced lines. So the range there was variation, however, it wasn't a lot.
But your graph was showing fairly large variation.
Well, that's because it was an area under the curve
Oh.
rating. It wasn't an actual, and that was at 3 different points taken.
Why I ask is maturity is normally correlated with yield. And if both are varying in same experiment, you will see what you are saying, 3 QTL from maturity, 3 for yield. So when we are looking for yield, maturity should be more or less similar among the progenies. Then you see the yield genes. Otherwise, it is maturity gene which is reflected in yield. So anyway.
Yeah, and we saw the co-location of the peaks for maturity as well as there was a yield, and we thought they're probably the same peak that we're looking at. There's probably the same genes.
So if you extend upon that, the candidate gene they pulled out was CDF1. Can you expand upon the candidate you had that was 1 megabase from that? And
Oh, the marker I found?
yeah, because CDF is a maturity gene or it's related to.
Yeah, it's a maturity as well as
What was the one you
tuberization.
found that was close to that?
It was an unknown gene in the database. It was but it was really, really close to that CDF1 gene. But we didn't have a marker for that CDF1 gene in our population. But yeah, it was right there.
So how would you respond to a reviewer with Stevie's question that CDF comment? Possibly all you're seeing is maturity.
I agree. I believe we wrote that in the paper. It's most likely the same QTLs. Because the yield, if you noticed, it was a wide range, and then everything else peaked within that range of yield. So we were attributing the yield not necessarily to a QTL4 yield, but every other QTL within that range, such as maturity, tuberization, virus resistance, internal root necrosis resistance. There's so many things going on there. But we were able to localize them to this one area. So we started to believe, hey, we need to look a lot into chromosome 5 in the future.
So you talk about the phenotypes weren't random and independent, but I don't think that's necessarily the only question, because if you have some type of strange perturbation in your map, you can also get significant effects at the same spot over years, because you're using the same map for all of those. So did you look at segregation distortion or missing data in that area?
For the markers?
The markers.
Well, we did a segregation analysis initially on the markers, and if they didn't pass chi-square with very stringent significant level, they wouldn't have been in the map in the first place. But no, I did not go back and look at after that point.
Because the permutation test is across, it's across all markers in the genome. So if that LODS permutation was set based on, say that whatever, I don't remember if it was chromosome 5 or whatever, if that was the marker that was causing the LODS, the permutation threshold to be as high as it was, and you're finding consistently over years that there's also QTL there, I would be really sketched out by that QTL.
Okay. So go back and check the distortion.
Yeah, or missing data, or I don't know if Dave Stelly's here. I can never remember the guy's name. There was he hosted somebody from Mexico who was looking at sort of information indices in each marker, and it's amazing how different each marker can be because of missing data and segregation distortion for how much information it provides for mapping the QTL. And so you can get a lot of false positives and negatives because, you know, just at a certain marker.
When you say missing data, you're talking about how many no calls that marker particularly had?
Yeah.
So, yeah, like I said, when we built our map, we removed markers that had more than 10% no calls within the progeny, and we did a segregation analysis at first, but no, I didn't go back and look at those particular markers after the fact. And I did a QTL analysis on the original data values. So we were hoping we had a good map, and but no, I did not go back afterwards.
It's just the assumption that it's independent random isn't just about your phenotypic data, it's also about your molecular map data, and those are not independent from year to year, right?
That's right. Yes.
You map 20 QTLs, right? And there are 4 years of phenotype data. How many of the QTL you map the same spot all 4 years phenotypic data? How many of that?
How many of the 20?
Yes, map the same location.
I don't know,
Not all.
but I did mention which ones had multiple years in the same location when I wrote it up. I don't know offhand.
What means multiple years? All 4 years, or 3 years, or 2 years?
For IHN, because there's a lot of variation in environment for susceptibility, we reported those that had at least 2 years, different years at the same location. But for instance, maturity, we had every single year at the exact same location. And some of the big QTLs we had every single year at the exact same location. So I reported those, and I reported the ones we found, but I did say that we're building a case for it. And there's obviously more evidence with these because there's multiple years at the same location than these that there's not evidence for this.
Yeah, but certainly something cross years, that's good one.
Yeah.
But also year specific may be good one too, maybe critical for the adaptation of the cultivars or plants to particular environment.
Absolutely. We did find in 1 year for internal heat necrosis, which was the hottest year, and we did find one QTL that stood out in that year only, and then we had, that was the year that it was almost drought, and we had the super high heat, but it only showed up in that 1 year. But we did say that, you know, this might be something because of the temperature and lack of rainfall this year. But yeah, you're right. Some years are going to be more favorable environments for identifying the QTLs than others. I wouldn't trust. And we didn't find too much in 1 year also because we didn't have a lot of results, a lot of environments that favored that type of effect from the plant.
All right, well, if there's no other questions.
Are you working on potato here also?
I'm working on cotton.
Orchard. Oh, you are here.
I haven't looked at the potato
You
in 5 months.
We have a very good potato program here.
How big of working on potatoes? Supposed to work on
You gotta work on something you
the
can't eat.
fabric of our lives.
He keeps digging up all the cotton plants. I don't know.
Something same.
Keep looking for something
Something
to eat.
same, all polychloride. All complicated polychloride species. Potato and cotton.
All right, well, let's thank Mitchell for his presentation.
Before everybody runs off, I'm just going to mention real briefly that we have a prospective student visiting, Rosalina Tinkofa, and so, Rosalina, you want to raise your hand? So any faculty members who are looking for a potential graduate student, she's from Peru. She has funding from the government for Peru, and she has expertise in bioinformatics. So, Thank you.